thank you for your attention today. As I said, I can use a little bit more of time. I hope to finish within 30 minutes. So this is unlike some of the previous talks in the sense that we're going to be doing some bioinformatics and computational stuff rather than applications and RNA transcriptomics. Although the results that you see presented here are applicable to a wide range of problems in both DNA and RNA. So life is wonderful, and the enormous variety of life is just amazing. It's extremely complex, as we were saying this morning, and we have learned in the last 30 or 40 years that molecular analysis is absolutely critical. And what we want to do is address this problem that we were discussing this morning of integration of data across spatial and temporal scales, very large, not only in terms of nano, meso, and macro levels, but also in terms of integration across time. So this has been accomplished. Luckily, I can use an analogy to explain what is going to happen, because something similar happened early on with what today we call GPS. I guess the story here is that 400 years ago in physics, people wanted to know how planets move, and they would collect data of several points, astronomers did. And the question was, how do the planets move? And of course, Newton figured out a way through gravitation to understand that 3 points allow you to determine the trajectory of a planet because it moves in an elliptic orbit. And so it was necessary to establish a coordinate system that allowed the determination of this trajectory in that reference frame, and the minimum amount of information necessary to determine the orbit. So today, jumping forward 400 years, we have GPSs that we use in our cell phones every day. So the idea then there is to provide a reference frame to locate any event in space and in time, and GPS offers exactly that about planet Earth. So what we want to do is something similar, but in a much more complicated sphere, biosphere. That is the sphere of life, this diversity and extension. So here's the talk. Is going to be 3 pieces. The first 2 are going to be very brief because they are very technical, and I don't want to burden you with the technicalities. But at least they'll give you a sense of the research that is at the foundation of the system. And then I will explain how the system works briefly, and finally provide a couple of applications that may illustrate to you how it could be used. So I work in a field called DNA computing, molecular computing. The purpose of which is to essentially build computers out of DNA molecules. So the conventional wisdom about computers is that they're made of electronics. But this time we want to use real DNA molecules and the molecules to build entities that process information the way ordinary computers do. So this was a field initiated about 20 plus years ago by Adelman of RSA fame, the cryptographic scheme. And this year we celebrate the 21st version of the conference where experts meet to discuss these issues. So the basic idea is that the native properties of DNA can be used to self-assemble computers. And I'm not going to go into the details of that, but here's the smoking gun that shows that such things are possible. They were developed by Winfrey at Caltech about 15 years ago or so. So if we want to use DNA molecules to process information, then we need to encode that information in DNA. And to encode that information, we have to convince or twist the arm of DNA to do things that it was not supposed to do natively, that is process information. So we have to select DNA very carefully to make sure that it doesn't do what it's supposed to do natively, but it does what we want it to do in terms of processing information. So this is known as the code word design problem. And the basic idea then is to select oligos of the sizes varying between 10 mers or very short n mers to 100, 150, 200 mers that will allow you to encode that information, let's say on a chip, on a DNA chip. So what we want to, if you select those probes, we're going to call probes what is affixed on the chip. Are we going to call targets what is put on the chip and what is to be processed by the chip to obtain some information. So what we want to do is obtain what is called, what I'm going to call DNA NXH basis. NXH stands for non-cross-hybridizing oligos. So these are oligons that have been very carefully selected so that they don't interact with one another, on the one hand. But more important than that, they provide full coverage of the entire DNA space. What I call a DNA space is, for a given size of resolution, the length of the n-mers in the probes, we're going to select a complete set of, let's say, for example, n is 20, so 20 mers. We're going to make a selection of 20 mers in such a way that every other 20 mer, first of all, they don't interact with one another through cross-hybridization. So in particular, none of them are complementary to any other. Second, they don't even have enough affinity to form a stable bond. And third, any other 20 mer is going to stick to exactly one of those 20 mers. So it's going to, in other words, this universal coverage in the sense that if I drop any 20 mer in there, or something that has an overlap of 20 mers, is going to give me a signal of some sort to one of those probes. So that's what we're going to call a basis. And as I said, they have universal coverage. So this problem, if you look at it from the computational point of view, is very difficult to, is equivalent to optimization problem, the trivial assessment problem, which is known to be very hard to solve on conventional computers. So, however, what we have done here as the basic research is to find a method to select these probes very carefully so that the corresponding chip will have the properties that I just described of complete coverage and universality. So that in other words, we have a solution to the code word design problem, which is very difficult conventionally. And we have done so here's an example of one such chip. I'm going to be presenting slides of this sort. White means non-cross-hybridizing, no signal, no attachment, whereas dark means full attachment. So this is a slide that shows is all against the other. So every probe, every probe is checked against the others, and the fact that it's basically white indicates that there's no cross-hybridization. So Gibbs energies have been normalized to 0 and 1, where 0 indicates no cross-hybridization. Minus 6 kilocalories per mole has been mapped to 1/2, and the minimum Gibbs energy below minus 6 kilocalories per mole indicates hybridization, stable hybridization. So as you see, most of them are not non-cross-hybridizing, although there may be this one here may be hybridizing to some of them. So these are not, these are good because they're mostly white. If you take a random sample of 20 mers, you're going to get something that is basically black. And so these are pretty good quality, although they're not the best quality we could provide. So once you have that basis, then I'm going to talk about DNA chips, although this is totally independent of that because most of us are familiar with microarrays, so they'll be easier to understand. So once you have your probes, then you fix them on a chip. In this case, we did an example of with 7 human diseases here. And that's 2, 4, 6, 7. We extracted from that a non-cross hybridizing subset and obtain, and we run the genes associated with those diseases, and they produce the signal that you see here. So in other words, to obtain a signature for a particular genome or gene, what you do is slice up the genes by ultrasound or something like that, or cleavage, into oligos or fragments of size roughly the same size as the probes. And then you create the appropriate chemical conditions so that they hybridize, they attach to the probes, and they provide a signal by the intensity of the probe. So here, one of these, for example, the lighter ones reveal a high signal, and the dark ones are a low signal. And so you see this an example of if you were to take all the possible fragments in all the genes together, and you're trying to differentiate the diseases, then you would see this kind of thing. So this is an example of the signals that we would obtain if we didn't use a carefully designed chip. As you can see, there, two of them are very hard to distinguish, for example. And there's a lot more probes up here. This is 121 or so probes, whereas there's only about 49 probes down here. And the point here is, these are some analysis of the signal-to-noise ratio. And the point is that with fewer probes that have been carefully selected to avoid cross hybridization, you get much cleaner and lighter signals. And we all know that microarrays are very sensitive to there's a lot of signal, and the signal-to-noise ratio is very low. So the first lesson that we learned from this system is that larger is not necessarily better. That putting the largest amount of probes on the chip is going to create a lot of noise, and here's a way to clean out the noise and provide better signals for what you're getting. So that's an example of a careful design, how a careful design provides you a lot more information, because it's much easier to distinguish two diseases here by this signature on that chip, on the clean chip, on the non-cross hybridizing chip, than you would get on an ordinary DNA chip or a microarray up here. So that's the first part. The second part is that we have found a much better way to design these chips so that essentially give universal coverage. This example here was done only for a peculiar design for those diseases. In this case now, we have been able to come up with a design that allows you, that gives you a truly universal chip that will provide a signature for anything, for any kind of set of oligos that you throw on that chip will provide a signature of some sort. And I don't want to go into the technicalities of this, but you might ask, how does this relate to real life stuff? And the upshot of it is that on this chip, the decision that this chip makes about a hybridization event agrees with what it would happen in real life as compared to the model, with a probability of 80%. In other words, this is not a perfect model, but it gives you about 80% of the truth that you will see. And of course, there's a trade-off. The model is a little bit off, but on the other hand, it affords some advantages that you wouldn't have otherwise. So these are the technicalities of the design of the chip, but to give you an intuitive idea of what's happening here, it boils down to packing. Designing a universal chip is a problem in sphere packing. All of us have packed our suitcases, and we know the problem, right? We know that if I put something here, I cannot put something else there. So and the man in the street actually has a pretty good idea how to do this packing. This man, as you can see, this man, what is he doing? He's trying to sell his fruit, right? And he's trying to maximize his exposure, right? So what does he want to do? He wants to pack the maximum amount of lemons and oranges and so forth into the minimum amount of space. And that's exactly the problem that we need to solve, The only difference is that that problem has to be solved in a very high dimensional space, not just three-dimensional space. But if you have 6 mares, you have to solve the problem in 24-dimensional space. If you have 20 mares, you would have to solve the problem in 80-dimensional space. And so here are examples. So this is a nice packing of pennies in a 2-dimensional space. That gives you an idea of the optimal solution. It's a hexagon. Believe it or not, it took 200 years for mathematicians to prove that that's the optimal solution. This is an example of cannonballs that are the optimal solution in 3-dimensional space. And this is an example of some phenomenon that illustrates the packing in 4-dimensional space, which would correspond to just simple ACGT. So we are trading off this optimality for reasoning in high-dimensional space. So that's just to give you an idea of what is involved. So fortunately, people have been working for the last 200 years on this problem, last 200-300 years on this problem, and they have found very interesting solutions. The ones I'm going to use today, to show you today, have to do with this so-called Leach lattice. So anybody who works in physics or even chemistry will know about the Leach lattice, because it has to do with crystallography and so forth. So this is a surprisingly nice packing that happens in 24-dimensional space that gives us a nice universal chip for 6 mares, for example. So that's just to, so this was developed back in the 60s and 70s to solve problems in communication theory, but we have been able to apply that to the design of the DNA chips here. So here are some statistics on the best known table that shows you the best known designs for 6 mares, 8 mares, 12 mares, 16 mares, 20 mares. So we have developed some procedures of evolution in vitro to obtain some chips, but they're not as good as the ones that I'm presenting today. So now that's the second part. Let's move on to the application part. How am I doing? I'm doing good. How much? Oh, great. Great. So let's see if we can talk a little bit more about the applications, because that's the juicy part that you probably want to see. So what is this chip good for? So I'm going to give you 2 applications that we're developing. One of them has been fairly developed. It was a PhD student 2-3 years ago, and the other one is a project. One of the major projects that we have today is the Tree of Life. And so I'll explain them in turn. So what is it that you want? I mean, this is the gadget. What is it that you want to do with it? Well, you want to extract information out of DNA molecules, right? In one way or another, either expression levels or correlation between expression levels, genomic analysis, transcriptomic analysis, and so forth. That's what you want to do with it. And so to give 2 proofs of concept how it is possible with this kind of gadget, I'm going to talk to you about semantic analysis. So some of you may have come across this. All of you have done semantic analysis in some form or another in using your Illumina, Pacific Bio, etc. It's exactly what you want to do. You want to do data mining. You can get all this data, and you go through all this trouble in the lab to get all this data. But if you can't analyze the data and get the conclusions that you want about your problems, all this is for nil. So semantic analysis has to do with that. So the idea of semantic analysis is that DNA can process information in a semantic way, in a way that you and I would do it. So semantic has to do with the idea that I don't really care how you represent the information in the computer. I care about what it means, or what conclusions I can get in human terms, in terms that you and I can understand and use. So in other words, I don't want data. I want information that I can use. So Baum demonstrated back in the early days of biomolecular computing that this is indeed possible. So he said, well, here's my database. It's a bunch of DNA molecules like my chip. It's like the chip that I just told you. It doesn't have to be on a chip. It could be in solution, because the molecules are not cross-hybridizing, so you can just toss them into the tube. And so this is a database of a company, for example, with all their customers. And each DNA strand encodes something like the customer's name, an ID, and balance, and whatever else. And you want to know John Smith. John Smith's record, how much does he owe, what is his account number, blah, blah. So what you do is drop a probe, like a fishing line, into that, and that contains the complementary strand to the one that contains the record for John Smith. And of course, there will be, if this complementary is going to be hybridization, and the fact that they're non-cross hybridizing will assure you that only that particular record for John Smith will be pulled out of this mess here. Of course, there could be billions of molecules in there encoding billions of customers' information, but only the one that is relevant to John Smith in some sense or another, because there's some affinity in that complement, will be pulled out. So then there you go. That's how semantic memories work. And they're semantic because although DNA is encoding information, it's not encoding information the way conventional computers encode it, that you have to have an exact match to be able to retrieve that based on an address from memory somewhere. The molecules are moving around and they contain the information. They're not pinned to a single address. They're just looking for the complement. And as long as the complement that presumably matches the information that is being requested, you're going to get only the right thing and the right thing, if it is there, of course. So that shows that semantic memory, that processes information the way we humans would, is possible to retrieve from DNA devices. So the best known processor of semantic information in electronics is called LSA. So LSA stands for latent semantic analysis. There's variations of it. It's an example of so-called single value decomposition and principal component analysis, if you wish. And that's what it is based on, but this was originally done for electronics. So here's an example of a particular semantic problem to make the point. It's called textual entailment. So what is it? What is the problem? The problem is you are given two texts, the T for the text and the H for the hypothesis. And the text essentially gives you a situation or a context, and the hypothesis gives you essentially a question. Is this true or not in that context? So in this case, the shooters escaped as other soldiers tried to give aid to the wounded. That's the situation, that's the text. And now you want to know whether this hypothesis is true based on that text. And the hypothesis is the gunman escaped as other soldiers tried to help the wounded. So if you give this to an ordinary computer, the computer is going to be befuddled by the fact that shooters and gunmen are different words. They are spelled differently, so they're considered to be different words by the computer. But you and I know that in this situation, shooters and gunmen are equivalent, right? They're the same thing in the context. So you and I will say, sure, the hypothesis follows from the text. But the conventional computer will say, gee, I'm not sure, because those two words are different. It doesn't have a sense of the semantic equivalence of the two. So it's important in that case, for example, to solve this problem of word disambiguation. How can the same word can have two different meanings, like the word belt here. And it's in this case, you need to, in semantic analysis, it's important to distinguish between the two. And then what my student did was to run a study using the DNA chips of the sort that I have described to you, and then using a word net, which is a network developed at Princeton by Miller in '85, to try to capture the idea of word knowledge. Because one of the things that we use in an inference, like solving the entailment problem, is we pull every bit of knowledge that we have in our head about gunmen and shooters to solve the problem, right? To decide on the equivalence. So by encoding that word net into DNA molecules, then here's the upshot of the results. So we were able to, by a very simple method of DNA hybridization, we were able to at least be competitive, if not better, than the best methods for textual entailment. This is a standard problem in semantic analysis, and there's a yearly competition where entrants use their solutions to solve a standard set of 800 questions of the type that I gave you. And there's winners and losers and all that. But here are the results that they got versus our results. And they're very competitive. So these are very sophisticated methods with a lot of computational intelligence and things like that. But we were able to match that performance based on simple DNA hybridization on this chip. So that gives you a sense how you can perform semantic analysis on these DNA chips using biochemical ideas. So the next example is the last example is the tree of life. This is some chart here about the power of along 3 axes in terms of speed, size, and semantic quality of a number of semantic analyzers. So I'm not going to go into that. The last part then is the tree of life. And so this is closer to home to most of you. So what is it that you want to do? Well, we have fossils and we have DNA, and DNA is a recorded history of evolution, that what's happened in the past to be what we are today. And what is it that you want to know? Well, given your genome, or the genome of a number of species, you want to decide. The phylogenetic analysis will give you some idea of the evolutionary paths that those organisms have followed through their DNA record to become what they are today. So a standard project in biology, in evolutionary biology, is to build a tree of life that gives us a sense of how life has evolved on planet Earth. So what we're doing is, the second proof of concept I want to show you is show how this kind of analysis is useful to build to do evolutionary to do phylogenetics and evolutionary analysis. So so initially we took 6 bacteria here on one of the good DNA chips that we designed, and then what we wanted to do was test the hypothesis that we can use the signatures that those genomes produce on these chips to build a phylogenetic tree for those organisms de novo, absolutely from scratch, in a way that is perfectly consistent with what biologists have elucidated in the course of 30 or 40 years of research to build more or less the same tree. And so that was exactly, so what you are looking at now is the signatures of the 6 bacteria on one of the chips that we have designed. And then here are the bacteria that we used, and this is more or less the size of the genome as downloaded from gene banks. And then we used only the open reading frames for that. And then we used, these are details on the chip that we got, but the upshot is, this is the phylogenetic tree that biologists have developed over the course of years, and this is the phylogenetic tree that we obtained using the chip. And as you can see, it's essentially producing the same phylogenetic tree, except for a small variation here that these 2 are swapped. And even that is debatable within this tree, whether that should be correct or not. So the point of all this then is that indeed the amount of information that you obtained essentially instantly from this chip is equivalent to in content to the information that you can obtain by other means from standard biological methods. So we repeated that's the upshot of that. We repeated the experiment with 17 bacteria, and we essentially got the same results. So we reproduced de novo the same analysis that biologists have obtained in by other methods, much more costly in terms of morphogenetics and so forth. So the tree of life that we are developing now, here's my last slide basically, or next to last slide. So what we are engaged now is in the Tree of Life project, which is trying to build an atlas of life based exclusively on the signatures that an arbitrary genome produces on, this on these chips. And here's an example. If you run the most elementary chip, is the one that has two values, the all A's and all G's, for example, and the signal that you'll get from there is the GC content of the organism, which is the simplest, most primitive kind of chip that you could get. On a more sophisticated chip, for example, here's an example. We can get the stop codons. You just look at the stop codons for a chip. These are the codons. And so we took 12 organisms representing 12 different, an exhaustive representation of the coarsest level of all life. And it produces these signals on that chip, which is still a very, very rough, very primitive chip. But even there you can see that this first probe, there are 12 probes, I'm sorry, 3 probes, these 3, and you produce the signals here. And then even that probe, that first probe, is an excellent marker to distinguish this organism from all the others. And so forth. So the fact that if you take a vector of the 3 signals coming from the 3 probes, you'll see that that's a complete set to distinguish amply each of the organisms from all the others. So it gives you, this is a very coarse chip. It's a very small, only 3 probes, and gives you that differentiation already. So the plan now is to enrich the chip and get a more and more sophisticated chip. And if all goes well, we'll be able to, at least in the case of the bacteria, we were able to differentiate the chips down to the order. And I think with a richer chip, we'll be able to differentiate down to the single organism, single species, and even maybe single organism if the chip is refined enough. So this is just beginning now. This method is absolutely general. If you want to use the chip to distinguish, it's a universal chip. So if you just take a single gene or a single segment of a gene, it will give you a signal of some sort. And the point is that on the same chip, you can compare everything. Everything, absolutely any DNA will give you a signal. And it's all on the common reference frame. So in other words, it doesn't matter if it came from a gene or a completely different chromosome or organism, it'll give you a signal of some sort that you can compare on the same frame of reference. So perhaps then we'll be able to determine the trajectories of life as Newton was able to determine the trajectories of the planets. So, thank you.